Thank you for joining us for our next session, Genetics, Genomics, What It Means and Why It Matters. Thank you to Genentech for making this session possible. Genetic testing, genomic testing, tumor testing, gene sequencing, what does it all mean? These terms are confusing, but important to understand. Dr. Barbara Norquist will break it all down for us. Dr. Norquist is a board-certified gynecologic oncologist and an assistant professor in the Department of Obstetrics and Gynecology at the University of Washington. Thank you, Dr. Norquist. I'll turn it over to you.
I'm Barb Norquist. I'm a gynecologic oncologist at the University of Washington in Seattle. And I've been asked to discuss genetics and genomics, what it means and why it matters. I have no financial disclosures. So the NCCN guidelines for the management of ovarian cancer specify that patients with ovarian cancer, fallopian tube cancer, or primary peritoneal cancer should have a genetic risk evaluation and both germline and somatic testing. And then throughout this talk, I'll use the term ovarian cancer as a shorthand for ovarian, fallopian tube, and primary peritoneal cancers. Considering that they tend to behave the same and are treated the same by oncologists. So what are germline and somatic genetic testing? So I'm going to spend some time on this because these terms can be fairly confusing. So germline testing implies testing DNA that was present essentially since birth. And these are looking for inherited mutations that are present in every cell in your body. This testing is done by examining either a blood sample, a cheek swab, a spit sample. And again, these are mutations that impact every cell in your body. Somatic testing implies testing acquired mutations in a body part. And so in this particular case, what this usually means is testing of the cancer itself or tumor testing, either from a biopsy or during a surgery. And this is looking for, again, these mutations that have occurred spontaneously in the cancer itself. Now, with the germline mutations that are present in every cell in the body, they will also be present within the cancer. And so if you test just the tumor, you might also find inherited mutations as well. So again, germline, every cell in the body, and then somatic are the cells within a tissue or a tumor. I'll go through just a few of the commonly used terms. So the term genetic testing, when it's used by medical providers, is most often used to describe this germline genetic testing for an inherited predisposition to cancer. And again, that's the blood or saliva testing. Tumor testing or tumor sequencing, you'll often hear providers refer to. This is also technically genetic testing. But it's usually used to describe this somatic genetic testing for acquired mutations within the cancer. And this is predominantly used to find predictors that can impact treatment. And again, this is from a biopsy or a surgery. The term genomic testing is sometimes used. The actual definition of this term is testing that involves the entire genome rather than individual genes. But most people use this as kind of a catch-all term to refer to multiple different types of genetic testing. So you might hear any of these terms used when discussing tests to get as part of an ovarian cancer diagnosis. So I'll speak now specifically about inherited risk and ovarian cancer. So inherited mutations in genes that can predispose to ovarian cancer are really prevalent in patients with ovarian cancer. This is from one study where we sequenced almost 2,000 patients with ovarian cancer, and we found that about 18% had inherited mutations that could have been the cause of their ovarian cancer. This is one of the highest proportions of any of the cancers. So what does this mean for a patient with ovarian cancer if they have one of these mutations? So BRCA1 and BRCA2 are proteins that work to repair DNA within the body. Tumors that have BRCA1 or BRCA2 that doesn't function properly can have a hard time repairing DNA, which can lead to the development of tumors or cancer. Somewhat paradoxically, this defective BRCA1 or 2 also makes the cancer much more susceptible to therapies that damage DNA, such as chemotherapy. PARP inhibitors are a relatively new class of drugs that specifically impact a different DNA repair pathway. So for cancers that have defective DNA repair, this can lead to the death of the tumor cells. It's kind of a double whammy. The scientific community uses the term synthetic lethality to describe that double whammy phenomenon, in case you've heard those words also. So in terms of what this means for patients with ovarian cancer, this is just one example of the impact that PARP inhibitors can have for patients with ovarian cancer, particularly those with inherited mutations or mutations in the tumor in BRCA1 and 2. And this was a study that looked just at that group of patients. So this is about 15% of all patients with ovarian cancer who have these inherited mutations. And they gave these patients maintenance olaparib therapy after they had completed their first round of chemo. And you may or may not be familiar with looking at this type of clinical trial data, but essentially these two lines describe the two different populations in the study. The blue line representing patients who were given the olaparib maintenance, and then the red line indicating those that had a placebo. And the amount of distance between the curves really points to how effective the therapy was. And in this particular trial, it was remarkably effective. And the bottom axis of this graph is months, and 60 months is 5 years. And so this is indicating that the patients who took olaparib, less than half of them have experienced a recurrence of their cancer, in contrast to those in the placebo arm who experienced many more recurrences. These drugs have also been studied in patients receiving PARP inhibitor maintenance therapy after treatment for a recurrence. And these are just several of the studies that have been published. And again, these big separations in the curves show you how effective the PARP inhibitors were in these groups. And again, these were all patients with BRCA mutations for the most part. These studies have led to multiple FDA approvals for maintenance therapies in patients with epithelial ovarian cancer. And this table just kind of shows the setting of the ovarian cancer, the population that it applies to, and the FDA-approved drugs. Olaparib, niraparib, and rucaparib are all PARP inhibitors. And then bevacizumab is not quite the topic of this talk, but it is also approved as a maintenance therapy in several settings. So we talked a bit about what having inherited or somatic mutations in BRCA1 or 2 can mean for treatment of the cancer. But a really important aspect of testing for inherited mutations is, what does this mean for the patient with ovarian cancer's family? This is a picture of a family tree. Um, this dark circle here indicates a patient with a BRCA1 mutation. Essentially, each of this person's first-degree relatives, so in this case, these would be siblings and children and parents too. Each first-degree relative would have a 50% chance of carrying the same BRCA1 mutation, and so genetic testing would be indicated for these family members who do not have cancer, so that they can undergo extremely effective methods of cancer prevention, such as screening for breast cancer and risk-reducing surgeries to remove ovaries and fallopian tubes, which is a highly effective way of preventing ovarian cancer. So a few words about how one inherits a mutation. So the BRCA1 gene, just to give a specific example, is located on chromosome 17, and each person has 2 copies of each chromosome. If the mom, in this case, has an inherited mutation in BRCA1, the subsequent child will either get will get one of mom's copies of chromosome 17, and that child will either get the one that has the BRCA1 mutation, or it will get the one that does not. Sometimes patients ask me if these mutations can be carried and passed along by male members of the family, and that is 100% true. They just won't manifest the cancers that are typically associated with a BRCA mutation. They can, but it's much less common. And so these genes and the mutations are not specific to gender. And so a father can definitely pass along a BRCA mutation to a child. Can mutations skip people in the family? I sometimes have patients ask me if they've had negative genetic testing, do their kids need testing? And the answer to that is no. It has to be directly passed from a parent to the child. So if this aunt has a BRCA1 mutation, but her brother is negative for that mutation, then these 2 kids cannot inherit the mutation. So what genetic testing options are currently available? Sometimes you'll hear the term single site testing or familial variant testing. And this is when you know what the mutation is in the gene. You can look just at that part of the gene to see if a family member has inherited that change. There are 3 founder mutations present in the Ashkenazi Jewish population that if a patient is known to have Ashkenazi Jewish heritage, sometimes they will check first for these 3 mutations. And then if those are not identified, then they'll broaden it to look for other mutations as well. The reason to do more limited testing in these circumstances is because it's cheaper and easier to look for just the specific changes than it is to sequence entire genes. Single gene testing has most typically been applied to the BRCA1 and 2 genes. So that's sequencing through the entire gene to look for a mutation anywhere within the protein structure. Gene panel testing, which is now by far the most common option, looks at multiple genes, BRCA1, BRCA2, and there's a number of other genes that can also lead to inherited predispositions to ovarian cancer. And so those can all be tested within one test with gene panel testing. This is sometimes also referred to as next generation sequencing. So how does one get tested for inherited mutations? The gold standard is really to meet with a genetic counselor. And this is a website where you can find one near you. They can often meet with patients either in person or by telephone. And now, since the COVID pandemic, increasingly these visits are happening through Zoom or through telehealth. The main advantages of working with a genetic counselor is they can help you help other family members, can help with insurance coverage, can really explain the testing and what it'll mean, and they'll order the most appropriate test for your situation. The main cons are that not everyone has access to genetic counselors. Sometimes insurance coverage of this visit can be inconsistent, and sometimes it places a barrier to getting testing. Another option is that your oncologist can directly order the testing and then have you see a counselor depending on what the results are. This tends to be faster, but the main con is you don't quite get the same pre-test counseling, and the oncologist might order the wrong test depending on their degree of knowledge about all of the different options. And so the optimal strategy for getting this done is really not yet defined, but there are multiple options. And then just a brief word about who else should have testing for inherited risk, at least per the NCCN guidelines. In addition to all patients with ovarian cancer, all patients with pancreatic cancer, patients with breast cancer who are also either young or have a strong family history, a bilateral cancer or a triple negative cancer, prostate cancer with certain high-risk features, people with known mutations in their family, like I was talking about before, which is also called cascade testing, and people who are unaffected by cancer but have a concerning family history involving these cancers. We are hopefully headed towards a future where all patients with any cancer will be able to have genetic testing. However, right now it's somewhat restricted, at least by insurance coverage for those populations that are listed in the NCCN guidelines. Another question I'm frequently asked is, well, I had genetic testing years ago, should I repeat it? And the answer to that question really depends on what year the testing was undertaken. And I'll show you just a very brief history of what types of testing have been available at what years. So in 1990, Mary Claire King discovered the BRCA1 gene. In 1995, a patent was filed for the sequence of the BRCA1 gene, and then shortly thereafter for BRCA2. So kind of the earliest a person could have had commercial genetic testing was in 1995, 1996. And then from 96 to 2013, one testing company owned the patents for the gene sequences, and that was the only way to get a commercial test. In 2009, next generation sequencing was developed, which allowed for gene panel testing to look at multiple genes at once. But this was not really offered clinically yet because of the patent restrictions. Then this all changed in 2013 with a landmark Supreme Court decision, which decided unanimously that the sequence of a gene was not an item that could be patented. This then led to an explosion of multiple companies, mostly offering gene panel testing. And so there are now many more options for how this would happen. And so if a person had genetic testing prior to 2013 that was negative, it is worth getting another test to see if any of the other ovarian cancer predisposition genes could be identified. These little asterisks also indicate where expanded testing of BRCA1 and 2 was initiated. So in 2006, rearrangement testing, which picks up about an additional 10% of BRCA1 and 2 mutations, came out. And then in 2013, it was done routinely rather than as a separate test. So I refer back to this timeline whenever I'm seeing a patient who's had testing at a particular interval, so I can figure out how much they might benefit from a repeat test. So now I'll focus on tumor testing. So again, this is testing typically from a biopsy or a surgery. There are some companies that that look for kind of circulating tumor DNA in the blood. That's called a liquid biopsy. And this is not necessarily what I'm discussing here. This is testing from biopsy or surgery. Sequencing is one of the main things that is done with these tissues, which is genetic testing, looking for acquired mutations in the cancer in BRCA1 or BRCA2, which are equally predictive of PARP inhibitor response, as well as other genes like RAD51C and RAD51D that can also predict responses to PARP inhibitors. So that is the purpose of that testing. And if you look at this pie chart here, this was a study done by Katie Pennington, one of my colleagues at the UW, where they found additional BRCA mutations in tumors in about an extra 8% of patients with ovarian cancer. So it's relatively common. Another type of testing you might hear about is called homologous recombination deficiency. And this is, again, looking at predicting response to PARP inhibitors. And I have a separate slide kind of describing what that is in a moment. Microsatellite instability, or MSI, is another term that might come up when a patient's undergoing tumor testing. This type of testing is more commonly useful in other cancer sites, like endometrial cancer in particular. However, if it is found in ovarian cancer, it can potentially predict responses to immunotherapy. And then you get a lot of other information about many genes and proteins within the tumor with a lot of this testing. And so I've just called that other stuff. Unfortunately, this is not super clinically actionable in most cases, but can sometimes impact clinical trial eligibility. So that can be a reason why that additional information can sometimes be helpful. So what is homologous recombination deficiency, or HRD? We can test for causes of homologous recombination deficiency, and this is that DNA repair pathway that I was talking about before with the BRCA genes. So inherited and somatic tumor mutations in BRCA1 and BRCA2 can cause homologous recombination deficiency, and inherited and somatic mutations in other genes that are in the same pathway as BRCA1 and 2, like RAD51D and RAD51C. And then epigenetic changes like methylation or other things that can impact the function of these genes. HRD testing, as it is looking for the effects of having a history of homologous recombination deficiency. And this history leaves scars in the genome that can be identified. It indicates evidence of past homologous recombination deficiency and does not necessarily indicate the current status. And that's why these tests of HRD, which look for the genomic scars, tend to be a little less predictive of PARP inhibitor response than something as definitive as a BRCA mutation. But it is potentially another thing that can be assessed that can sometimes predict response to PARP inhibitors. And I think of the HRD testing kind of as looking for the graffiti that HRD was here at some point. So another question I'm sometimes asked is my first tumor, so my initial cancer was tested with sequencing. Should I have repeat testing at the time of a recurrence? And I would say right now in 2022, This is most likely to just be additional cost and probably not terribly helpful for treating your cancer. Major drivers of drug response like BRCA mutations, microsatellite instability, and HRD don't tend to change on a repeat biopsy. Because again, HRD is kind of just that sign that that change was there at some point. It doesn't necessarily indicate that it's still present. Exceptions to this include research studies that are trying to predict responses to therapy and are following patients to see what happens to them, and clinical trial eligibility based on testing. So those could be some reasons to get testing on a biopsy at the time of recurrence rather than testing the initial tumor. And so hopefully as more scientific knowledge about this is gained, this type of testing will be more helpful in the future. But right at this moment, it's of less clinical utility. So if you're going to go through the expense and discomfort of a new biopsy, just be really clear about what the potential benefits might be. So here are my conclusions. So in summary, the standard of care for ovarian cancer is to have both germline and somatic genetic testing. So testing of for both inherited mutations and testing for acquired mutations in the cancer that could impact treatment response. These findings can help your oncologist treat your cancer, and they can help your family avoid cancer if inherited mutations are found. I would encourage you to save copies of any genetic testing reports. Most medical records are not designed to keep this in a place that's easy to find. And so this can be really critically important to keep track of. All right, so thank you so much for your time.